Deep in the Egyptian desert, an archaeological team pried open a clay jar that had sat untouched for 3,000 years. Inside these intact, perfectly sealed ceramic vessels, sitting at the bottom of a tomb older than the Roman Empire, was a pool of golden honey. It looked exactly the way honey looks when you crack open a fresh jar today. There was no mold, no fermentation, and the substance remained completely edible. This level of geological preservation is an anomaly. A piece of bread left on a modern kitchen counter will grow a visible culture of rot in about 3 days. Yet this honey survived 3 millennia without refrigeration, vacuum sealing, or industrial preservatives. We can dismiss mystical explanations or fairy tale ideas about a magical blessing from the bees. Biology operates on strict chemical rules. The shelf life of honey is dictated by a specific 4-part biochemical defense system engineered into every drop. Honey functions as an active, aggressively engineered state of absolute microbial stasis. The process begins with raw flower nectar, a fluid composed of up to 80% water. To transform this into honey, the bee colony performs a mechanical processing step. Thousands of bees fan their wings over the nectar to evaporate the vast majority of that moisture. The resulting chemical baseline is an extreme concentration of roughly 80% sugar, primarily fructose and glucose. High sugar concentrations exert immense osmotic pressure, which causes the sugar to actively seek out and pull in surrounding water from anything it touches. If a bacterium enters this environment, the water inside its own cellular fluids immediately rushes outward across its cell membrane to dilute the surrounding sugar. This forceful, fatal extraction of internal water is a process called plasmolysis. The invader dries out from the inside. Its cell membrane shrivels, its internal proteins denature, and the entire cellular structure collapses inward. You see the macroscopic version of this in your kitchen when you salt cucumbers or macerate strawberries to forcefully extract their internal juices. Honey achieves physical desiccation by pulling the water out of any organism that attempts to colonize it. To understand the next layer of preservation, we have to look past the total volume of moisture and measure what food scientists call available moisture. This horizontal scale illustrates a metric called water activity, written as AW. It runs from 0 to 1. Pure distilled water sits right at the 1.0 mark. For context, a bone-dry cracker plots near the bottom of the scale at 0.1. Look at the 0.9 mark. This is the critical biological threshold. Most bacteria and molds require a water activity above 0.9 to survive. The zone below it is entirely hostile to life. Even in its thick, syrupy state, honey still contains between 15 and 18% total water. Yet honey's water activity measurement falls far below the survival line, landing specifically between 0.5 and 0.65. This presents a paradox. How can a fluid containing 15% water be biologically drier than a cracker? The answer lies at the molecular level. Tight hydrogen bonds permanently lock the water molecules to the overwhelming volume of sugar molecules. Because these chemical bonds are so incredibly strong, the water is permanently trapped and unavailable for any microbial biological function. A microbe trapped in liquid honey effectively dies of thirst. The water exists, but it cannot be used. The next layer of the defense system relies on extreme acidity. Honey sits tightly between 3.0 and 4.5 on the pH scale. For reference, that makes it more acidic than tomato juice and puts it in the same neighborhood as vinegar. This severe drop in pH is driven primarily by a specific chemical compound called gluconic acid. Imagine an extremophile bacterium that somehow survives the initial osmotic crash and the total lack of available water. At a pH of 3, the internal biology of that microbe fails. Its critical internal enzymes begin to malfunction almost immediately. Simultaneously, the acid attacks from the outside, degrading the bacterial cell wall until it loses structural integrity and leaks. This is the exact same chemical process used in pickling, where acid kills off spoilage organisms to preserve vegetables. Honey functions as an internal canning factory, chemically pickling invaders from the inside out. Beyond these passive barriers lies an active, reactive biological mechanism. This layer is a direct biological additive injected into the mixture by the honeybee. That additive is an enzyme known as glucose oxidase. Under normal conditions, this enzyme sits completely dormant and inert within the syrup. It acts as a chemical tripwire. The enzyme instantly awakens the moment it comes into contact with external moisture, such as the wet cell wall of an invading microbe. When triggered by that moisture, the enzyme fires up and metabolizes the surrounding ingredients. As shown here, it takes glucose and oxygen and processes them into two new outputs. Those outputs are more gluconic acid and hydrogen peroxide. A 2011 lab study measured this output at between 0.248 and 2.68 millimoles of hydrogen peroxide per liter. This localized, slow drip dosage is highly effective at shredding a DNA of tough pathogens like E. coli. This enzymatic drip is why specialized honey is utilized in hospitals for treating antibiotic-resistant wound dressings. Honey is a reactive weapon system that strategically conserves its antiseptic, deploying it only when triggered by a target. These layers operate as a single interdependent system. The sugar enforces a drought, the drought slows the invaders, the acid disrupts their biology, and the peroxide executes the survivors. The clay jar provided a physical shield for this internal chemical system, insulating it from the outside world for 3 millennia. However, this biological fortress has one fatal flaw: external water. Everyday kitchen mistakes, a wet spoon or ambient condensation, introduce enough free water to destabilize the entire matrix. Adding that moisture pushes the water activity above the critical 0.6 threshold. Once that specific mathematical failure point is finally crossed, the structural integrity of the fluid completely breaks down. Dormant wild yeasts trapped in the liquid finally wake up and begin the process of fermentation, transforming eternal honey into mead. But as long as it remains sealed, the mechanism is immune to the passage of time. It functions identically in an ancient Greek hive, an Egyptian tomb, or a modern pantry. A jar of honey has no internal expiration date. It is an eternal, insect-engineered chemistry experiment that runs endlessly as long as the seal remains unbroken.